welcome everyone. Today we'll be discussing about biomolecules, chapter 9, class 11, biology and CRT. So when we talk about biomolecules, so there's a wide variety or diversity of living organisms in our biosphere. So there is, the components which have carbon, hydrogen, and all those things. These are the biomolecules which is required by the body. And how to analyze chemical composition? So if we look at any component, we can understand what is a chemical composition. We can know what is the chemical formula. So how can we analyze? We can have filtrate. Okay, say for example, we can take a living tissue, say a vegetable or piece of liver, and we can grind it in trichloroacetic acid using mortar and pestle, and then we will get a slurry. So this can be filtered out. So once the filtrate is there, the acid soluble pool, and second the retinate is there, or the acid insoluble fraction. So there are two fractions. So scientists have found thousands of organic compounds in this acid soluble pool. So herein we can have the formulas, we can get the molecular formulas of this, and all carbon compounds which is there in the body of a living tissue, they are the biomolecules. So we have the biomolecules. We are not talking about inorganic salts, but the ones which contains carbon. Okay, so one can weigh the amount of living tissue and try it. So all the water evaporates, and the material which is dry, that can be weighed. And once it is removed, water is removed, and the remaining is the ash. Once it gets burnt, evaporated, and all those things. Okay, so now there are different kinds of biomolecules out there. Like uh, we have got saccharides, that's carbohydrates. We have got proteins. We have got amino acids. We have got lipids. So all of them are there. Okay, so if I talk about amino acids, okay, if we talk about amino acids, there are 20 amino acids. There are so where the amino acids, they are organic compounds containing an amino group and an acidic group as substituents on the same carbon, that is alpha carbon. And then if we have a methyl group added to it, there's R, and based on the R there are several amino acids, like there are 20 amino acids. Next we have the lipids, which is water insoluble fatty acids. Okay, so that is there. So here the R could be methyl, ethyl, and all those things that is happening. So if we see that there is a Zwitter ion, what is a Zwitter ion? Which is having a partial positive and partial negative. Okay, so here is the Zwitter ion. Fine. So next, if we talk about, we have got DNA, RNA, which are the nucleic acids that we have. Okay, so here we can see the carbohydrates, which is the glucose, ribose. These carbohydrates are there, which has carbon, hydrogen, and oxygen. Amino acids like glycine, alanine, serine. These are there. Then we have fatty acids. So fats or oils, which is lipids, cholesterol, all these things. We have nitrogen bases, purine and pyrimidine, that is nucleoside and nucleotide. And then we have the nucleic acids. Those nucleosides, we have the adenosine or uridine, and with nucleotide we have adenolic acid. So this is the basic structure. Fine. All right, now coming to the Nexus, primary and secondary metabolites. So what is primary and secondary metabolites? So herein, what is happening, the most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms. Okay, so there are metabolites, so which is produced by the plants. So there are metabolized amino acid, sugars, and all those things. In animal tissues, one notices the presence of such categories of the compounds. Now, these are the primary metabolites like morphine, glutein, ricin. So, all these are the primary metabolites, but there are secondary metabolites which, like this, all this, they are the secondary metabolites. So they are produced by the plants, but they are not utilized. So they are utilized by human beings, so they are known as secondary metabolites. Well, primary metabolites have indefinite in identifiable functions and play an important role in normal physiological process we do at the moment. Understand the role of functions of all secondary metabolite in host organism. Next, we have the biomolecules. Now, let us come to the biomolecules, biomacromolecules, the large molecules that we have. So, biomacromolecules, when we talk about, we have got the acid insoluble fraction that has four types of organic compounds: protein, nucleic acid, polysaccharides, and lipids. So all these are there, and the macromolecules in insoluble, which with the expansion of lipids and are polymeric substances. Okay, now if we talk about lipid, they are small molecular weight, and we have cell membrane and all those which are broken into pieces. They form vesicles which are not basically water soluble. Coming to proteins, so in proteins what happens, there is amino acids. So these amino acids, when put together, they form the primary structure of the protein. These will have the hydrogen bonds, and it will form the secondary structure of the protein with the beta sheets or the alpha helix. Then we have the tertiary structure of protein because of van der Waals forces, hydrophobic bonds, sulfide, disulfide bonds. All those forms, they forms a the form a subunit, and when all the subunits comes together forms a functional protein, then it is the quaternary structure of the protein. So there are dietary proteins, there are many kinds of proteins which is there, enzymes also. Next we have the polysaccharides. Polysaccharides are basically the sugar molecules. So the starch which helps in storing a food, there are polysaccharides. So if single unit is there, then it is monosaccharide. 2 units disaccharides, oligosaccharides 2 to 9 units, and we have polysaccharides which are more than 9 units. We have nucleic acids, which is the DNA or RNA. The double helix structure of DNA was given by Watson and Crick. So that is the structure of the nucleic acid. Then here we have the structure of protein that I was talking about. You see the primary structure of the protein, which undergoes the alpha helix and the beta sheet structure. So the alpha helix will be in this way and beta sheet will be in this format. So they will be in the formation of sheets. Okay, fine. Next we have, so they will undergo the there will be the disulfide bonds, there will be van der Waals forces, so they will form the tertiary structure of protein, and then all they will come together and they will form the tertiary or the quaternary structure of protein. Okay, so this is how proteins work. Okay, now nature of bond linking the monomers in a polymer. So in a monomer to polymer, there are various bonds. So in a polysaccharide, I was talking about, if the same units of sugar which is joined together, they are homopolysaccharides, and if it is different, if the subunits are different, then they are heteropolysaccharides. Okay, so now here you see in the nucleic acid, so this is a DNA. So DNA, the nucleic acids, they are joined by a phosphodiesterase bond. Okay, so there is this kind of bondation is taking place in nucleic acids. So thymine is connected with guanine, and whereas cytosine is connected with adenine. So you see, and this adenine adenine and thymine is connected by 2 bonds, and guanine and cytosine is connected by 3 bonds. So this is how the structure happens in a DNA. That is how they are bound together. So between one, that is in one loop, it is basically 3.4 nanometer, and within that there are 10 base pairs. So that for one base pair, the length of one base pair is 0.34 nanometer. Okay, fine. Next we have the dynamic state of body constituents concept of metabolism. Now here we have that one of the greatest discoveries ever made was observation that all these biomolecules has a turnover. So this, what does this mean? This means they are constantly being changed into some or other biomolecule and also made from some other biomolecule. So thus there is a breaking and making which is always taking place. So these together forms the metabolism. So it has anabolism which is building up, catabolism which is breaking down. Okay, so that kind of bonds which are put together, they're catalyzed, okay, and or they can be broken down. So metabolic basis of living, so we need energy. We get the energy from ATP, adenosine triphosphate. So what happens here? So the metabolic pathways, they can lead to more complex structure from a simpler structure, or lead to simpler structures from a more complex. So when it is becoming simple to complex, okay, so pathways which leads to more complex structure, so simple to complex. So I said you anabolism is building up. So anabolism is building up. So okay, so here I was talking to you, anabolism is building up. So from simple it is forming complex. So this is anabolism, and the opposite is the catabolism. Okay, so what happens? So ATP, so ATP is broken down to ADP plus PI. So the catabolism is occurring, that is releasing out energy, and this ADP is further broken to AMP, at adenosine monophosphate plus PI. While building up, it is the opposite way, the anabolism which is taking place. Okay. Next, coming to enzymes. So when we have the enzymes, so these enzymes, they have an active site. So the enzymes, they have an active site. So there is an active site, the substrate also will have an active site. So they will match and they will come and bind. So enzyme plus substrate, it will form an enzyme substrate complex. These in turn will form the enzyme plus product. So enzyme basically is not taking part in the reaction but helping for the reaction to go ahead. Okay. So there are various chemical reactions which are there. There are various kinds of enzymes which is there. Okay. So next, coming to the chemical reactions. So there is a rate of reaction, because it is a rate of reaction, that is the change in the product formation with the per second, so per unit time. So that is the rate of reaction which is there. Fine. And now we have how enzymes bring about such high rate of reaction. So here you see what happens is, if I plot the energy, that is potential energy that the enzyme has, with that of the progress in reaction, what I will have, I am having a substrate and I am having the enzyme. So what it is happening, so substrate is increasing, it is reaching an activation stage. Okay, and then also the energy increases of the enzyme also, and it reaches a transition state. Then it goes, so enzyme and substrate, so their energy is increased and they reach an stage and they form this enzyme substrate complex. they form this enzyme substrate complex, and once this enzyme substrate complex is formed, it forms the enzyme product, and thus enzyme is released and the product is being released. So what are the factors affecting the enzyme activity? Temperature and pH. So temperature also, as the pH increases or as the temperature increases, the rate of reaction will increase, and after it reaches the activation state, the activity will keep on decreasing. Okay, so that is there. So activity of enzyme is basically inhibition. When binding to a chemical shuts off the enzyme activity, so that is known as inhibitor. So there can be two kinds of inhibitor. One is competitive inhibitor and a non-competitive inhibitor. So when I talk about a competitive inhibitor, what happens? The active site, so substrate is also having an active site and the inhibitor is also having the same active site. So what is happening? Inhibitor is coming and binding, there is a competition between both. Inhibitor is coming and binding, is not letting the substrate bind. And what is non-competitive inhibition? There are two sides, one for the substrate and one for the inhibitor. So the inhibitor will come and bind and the substrate will come and bind. Once the inhibitor comes and bind, it will no more form the product. Okay, so this is non-competitive inhibition. Now the reactions that takes place in an enzyme is given by equation, which is V is equal to Vmax S by Km plus S. So what is it? So V is the velocity. So if I plot a graph of the velocity and the concentration of the substrate that is taking place, so this might be a graph that is taking place. So now wherever this is Vmax. So I will take half of Vmax, which is Vmax by 2, and I will take it and I will take it here. This concentration, this is Km. Okay, that is Michael-Menton's constant. So this is how I get the V, velocity of or the rate of reaction. Fine. So there are various classifications of enzymes like oxidoreductase, transferases, hydrolases, lyases, isomerases, and ligases. So they have got their own functions like oxidoreductase. These enzymes, they help in oxidation and reduction. Transferases, basically that will transfer the substrates. Hydrolases for hydrolysis of esters, ethers, and all those. Lyases, which is removal of a group. Isomerases, which is interconversion between the isomer. Ligases is basically linking together the compounds. Then we have the cofactors. So enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called as cofactor, they will go and bind to the enzyme to make the enzyme catalytically active. Okay. So there are three kinds of cofactors. One is the prosthetic group, one is the coenzyme, and one is the metal ion. So prosthetic group is which? Prosthetic group, they are organic compounds and are distinguished from the other cofactors in that they are tightly bound to the apoenzyme, that is enzyme which is not stable. So for example, the peroxidase and catalases which catalyze the breakdown of hydrogen peroxide to water oxygen, heme is the prosthetic group and it is a part of the active site of the enzyme. Then we have the coenzyme. What is coenzyme? Coenzyme is again an organic compound, but that association with apoenzyme is transient, usually occurring during the course of catalysis. Further, coenzymes have several cofactors in a number of different enzymes catalyzed reactions. Okay, so there are a number of enzymes require metal ions for the activity, which is the cofactors which is there, and then catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. So that's it. Thank you.